in Cornwall, also on the round goby. So freshwater ecosystems play a really important role in natural environment. For example, they contribute to the global biodiversity, but they are challenged by many anthropogenic stressors like invasive species, but also like eutrophication. When we're talking about invasive species, we can think about the different impacts they can have on freshwater ecosystems, impacts that they can have at different level. For example, the community level, where they can modify trophic links in the food web, they can compete with native species. But also we can look at the ecosystem level, especially looking at energy flow. So for example, in a food web, you can have shift between pelagic and benthic energy pathways, and also nutritional quality of food web. And for this research, I'm focusing on the energy flow impacts of invasive species can have on energy flow. And when I'm talking about nutritional quality, I'm referring to essential fatty acids specifically. So these are essential fatty acids like the polyunsaturated fatty acids, so the PUFAs, especially the long-chain one, like EPA and DHA, which are omega-3s, and these are fats containing more than one double bond. And these essential fatty acids are synthesized de novo by phytoplankton. And different phytoplankton can have different essential fatty acids. Some will have more, and some others will lack those fatty acids, like cyanobacteria and chlorophyte don't have any EPA or DHA. So top consumers like fish will need to get their fatty acids mostly from their diet. And it's important for them to get essential fatty acid because it supports many things, including fecundity, reproduction, the metabolism function, and also some cellular structural functions as well. So essential fatty acid can be used as a tool for evaluating nutritional quality, but also body condition in fish. So invasive species can impact nutritional quality in food webs, but also can eutrophication. So an excess in phosphorus in aquatic ecosystems can promote a shift in phytoplankton community, which can be dominated by cyanobacteria. And cyanobacteria, like I said, are lacking those essential fatty acids. So eutrophication has the possibility to downgrade the availability of those essential fatty acids in food webs, and this effects can cascade up to top consumers like fish and affect their body condition or nutritional value. I'm interested in two specific invaders in this research, so the round goby and the Dresden mussels. So like it was mentioned, the round goby and the mussels were introduced in the Great Lakes in the 90s, and now they're spread everywhere, including in the St. Lawrence River and the upper St. Lawrence River. The round goby is a benthivorous fish, and Dresden mussels are known to filter water at high capacity. The reduced phytoplankton biomass, the increased benthic primary production, and they actually changed energy flows from pelagic to benthic pathways in the Great Lakes. So now, in invaded food webs, it looks a little bit like this, where you have the round goby in the middle feeding on native macroinvertebrates, but also feeding on the invasive Dresden mussels, which is not a prey that is typically exploited by other benthivorous fish like the small yellow perch, especially in the St. Lawrence River. And now piscivorous fish like smallmouth bass especially are feeding on the round goby. And a lot of research in the Great Lakes have shown that the round goby is actually a low quality prey compared to other native forage fish. And so, this raises the question as what is the impact of the round goby and the link with the mussel on the overall nutritional quality in the food web. So with that in mind, I had two objectives. So the first one was to investigate how the Dresden mussels and the round gobies modify the energy pathways and the nutritional quality, and whether these changes cascade to native piscivorous fish. And here I'm going to focus on the smallmouth bass. And secondly, to assess whether these invaders can alter the eutrophication effects on those essential fatty acids in the food web. So for the first objective, my hypothesis was that, the presence of those benthic invaders would enhance the benthic energy pathway in the food web, and that would result in low PUFAs, a low essential fatty acid, in the piscivorous fish, the smallmouth bass. Generally, the benthic pathway will lack DHA compared to the pelagic pathway, which can have a higher DHA to EPA ratios. And plus, well, the Rango bee is known to be of a low nutritional quality prey, so that's why I'm expecting lower PUFAs in the higher benthic pathways situation. Now, the second objective, my hypothesis was that the effect of eutrophications would extend from the cestins, from the phytoplankton, to the invasive Rango bee and the native small yellow perch that is also a benthivorous fish. And that would be resulted in low essential fatty acid as the total phosphorus concentration increases. So my study area is the Upper St. Lawrence River. So I used a space for time approach to assess my questions. So the Upper St. Lawrence River, like you can see here, is comprised of two major, in this section anyway, two major fluvial lakes. So Lake St. Francis and Lake St. Louis. And also connects to other fluvial system like the Ottawa River, which extends to Lake Two Mountains. The water quality in the Upper St. Lawrence River is influenced by agricultural runoff and also wastewater discharge. So there is some variation in the total phosphorus concentration, like you can see here with the orange to yellow color, with yellow being low concentration and orange being higher concentration. And the Upper St. Lawrence River is a hotspot for invasion because it's connected to Lake Ontario and the Great Lakes. And we can see variation in the Rango bee and dress and mussels abundance throughout the river. And because there is variation in connectivity, and that the Rango bee and the mussels are limited by low ion calciums and low connectivity, well, in this system, we find that there's no Rango bee in Lake Two Mountains because the water connectivity is very low. And there's higher abundance in Lake St. Francis and lower abundance in Lake St. Louis. So this is a perfect system for me to address my research questions. So I did a really big field sampling, 3 seasons. I sample across the food web from, the habitat, the water quality up the way to the top consumers, like my piscivorous fish. And on top of using essential fatty acid, like I explained, as a tool for evaluating nutritional quality and body condition in fish, I also use stable isotopes as a trophic indicator. So with the carbon-13, I can determine the diet of those fish because the consumers will reflect the signals of their prey. So I took muscle samples, which allows you to have a portrait of the diet between 2 and 3 months. And then the nitrogen 15 allows you to determine the trophic position. So usually consumers will be enriched in nitrogen 15 relative to their prey. Now let's move on to the results for the first objective. So here you have 3 panels showing the trophic position in Y in relation to the pelagic diet proportion. So here you have Two Mountains, which is the uninvaded lake, so no invaders. St. Francis with high abundance of invaders, and St. Louis with lower abundance. And you have the circle, which is the mean point for the smallmouth bass. Triangle is for the larger yellow perch, so above 100 millimeters. The diamond is for the smaller yellow perch, and the square is for the Rango bee. So what you can see here is that in the uninvaded lakes, those fish are more towards a greater pelagic diet proportion compared to, for example, here in the higher abundance of invaders, we have a lower pelagic diet proportion. So what we can say here is that the presence of those invaders promoted the benthic energy pathway. And we can explain those findings by thinking about the dress and mussels, that they filter a lot of water, they reduce water clarity, and now there's a higher benthic productivity. But there's also, the mussels creating beds, which can support higher invertebrate abundance. And on top of that, those piscivorous fish are feeding a lot on those benthivorous fish. So my second question was, now that we see there's a change in the benthic energy pathway, does that reflect in the smallmouth bass essential fatty acid profile? So what you can see here is a principal component analysis showing the bass essential fatty acid profiles. So in green, you have two mountains, so uninvaded. In orange, you have San Francisco, and in blue, you have Lake St. Louis. And what you can see is that those bass in two mountains have a distinct essential fatty acid composition. And the bass in the invaded lakes have markers, benthic markers of like the 1617 you see here in 1819. These are benthic feeding markers. Now, so I showed that the smallmouth bass have a different essential fatty acid composition, but they also have different proportion of those individual essential fatty acids. So here you have the DHA here in relation to the three lakes again, and you have a significant difference between Two Mountain and Lake San Francisco. So smallmouth bass have a lower DHA percentage in invaded lakes, well, the invaded lake. So let's move on to the results for the second objective. So what you see here, this is the cestin, and this is the proportion of the EPA in the water. And this goes from Kingston to Trois-Rivières in the St. Lawrence River. So the blue means low proportion of EPA, and red means higher proportion of EPA. And so what we can see here is between Cornwall and Montreal, which is where my lakes are situated, we have a very low proportion of EPA in the cestin. And this can be explained by the total phosphorus concentration. There's actually a significant negative relationship between the EPA and the total phosphorus concentration. So as total phosphorus increases, there's less EPA in the cestin. And this is probably caused by higher cyanobacterial abundances within the phytoplankton community between Cornwall and Montreal. Now, I wanted to know, well, does that extend to the invasive round goby? And what I can see is that, yes, so there's a negative effect of the total phosphorus on the PUFAs in the round goby. However, I didn't find any significant effect on the native small yellow perch. So what we can say here is that the effects of the eutrophication is mediated by the invasive goby and not by the native yellow perch. And the main difference between those two species, I think, is that the round goby will feed on invasive dress-in mussels, which are directly connected to the cestin because they filter the water. And the other explanation is that the round goby has a evolutionary history in brackish water. So just like marine species, maybe they're not able to do any bioconversion of essential fatty acids, whereas freshwater species, in some freshwater species, some can do at some extent some bioconversions. So just to conclude, so I showed that the round goby and the dress-in mussels promoted the benthic energy pathways in the upper St. Lawrence River, and that was reflected in altered essential fatty acid composition and reduced PUFA's percentage in smallmouth bass. And this is really important to point out because this could have potential consequences on bass reproduction or lifetime fecundity. So I think it's important maybe to focus future research on this because it could have implication on recreational fisheries, for example, and overall bass population health. Eutrophication, well, the invaders can accentuate eutrophication effects in food webs. So it's important to do research which assess multiple stressors at the same time in order to understand better and better manage invaded ecosystem. So with that, I'd like to thank everyone who helped me throughout my research and the funding throughout my PhD. So I will take any questions. Thank you.
Thank you so much, Cristina. That was really interesting. We do have a question Since round gobies are 15% lipid, does this mean that they have lower nutritional value because they're lower in essential fatty acids?
Yeah, so there's 15% lipid. I know, when you're talking about nutritional quality, there's other aspect to that, right? There's protein content, there's vitamins and things like that. But even within those 15%, it's important to have those higher level of essential fatty acid for the consumers. So I would say, yeah, they're the lower nutritional quality.
Okay, thank you. There's another question, and I can't really pronounce this very well, but I'll try. Have other piscivore fish been studied to see their fatty acid impacts from eating goby?
Yeah, so personally, I also looked at large yellow perch because they're known to eat gobies, but I didn't see the same results. So there was no significant effects of lakes on those yellow perch. And my guess is that although they eat round gobies, they have a very generalist diet compared to those smallmouth bass. So maybe that's why there was no differences in the essential fatty acid in the yellow perch. And also they live in various habitats compared to the smallmouth bass, right? Smallmouth bass will live rocky, in rocky habitats where the gobies are. So it's really easy for them to prey on those gobies compared to perch where they like vegetated habitat and things like that. So personally, I looked at perch and I didn't see that. But it would be interested to see for like pike or walleye that also eat goby if we see the same findings.
Yeah, awesome. Thank you. That looks like all the questions. So thank you again, Cristina, for a great talk.
Thank you.
Um, and thank you to all our speakers from today's session and our very engaged audience. This concludes the aquatic invasive session, and you can join us after lunch to hear from our keynote speakers, Jamie and Ross, who will be giving a presentation all about the impacts of aquatic invasive species. So I look forward to seeing you all there. Thank you so much.